And just as a quick, I think a quick announcement. We just started launch the new DGL version 9.1 about 10 days ago, like 2 weeks ago. So there's actually quite a few enhancements that we have added. One of the most notable enhancement we have added is actually just creating a new pipeline process data chunks for parallel training. So this is actually something that we are starting to aim at as an enterprise related service, and also for the academic, if you're interested in doing for the large graph training related training, it's definitely worth a look, and I would highly recommend the folks at the group to check it out, and I'll just share the links in the chat. Okay, and for our today's speaker, it will be Yusuf Roohani. So please excuse me if I'm not pronouncing the name correctly. So, but we have a good great topic today. It's actually regarding on the predicting the transactional outcome of the novel multi-gene perturbation. So I would, without further ado, I will give I think the presentation table back to Yusuf, and you can start.
Okay, great. Thank you. Hi, and thank you for attending my talk. My name is, as he mentioned, is Yusuf Roohani. I'm a PhD student at Stanford University. I'm really grateful to the organizers for inviting me to speak here today. Let me turn on my pointer. Okay. So I'll be talking to you about a project which we call GEARS, where we were interested in predicting transcriptional outcomes of novel multi-gene perturbations. So before we get started, I thought it would be useful to first just do a brief primer on some biological concepts, because these will be coming up a few times in this talk. So one of the central principles in biology is something that they call a central dogma, which is the link that goes from genotype, or your DNA, to the phenotype, or the actual physical traits that we have as living organisms. So all of us as living organisms have DNA, and DNA can be thought of essentially as like a collection of genes. And these genes are transcribed to produce RNA, and the RNA then goes on to produce proteins. And I'm sure most of you are aware that proteins are like the main functional building block of living systems. They're the ones that perform all the important functions. So this link is very critical in many biological processes. And for this talk, we'll be focusing mostly on RNA, and this is also referred to as gene expression. And the reason it's called gene expression is because you have many genes in your DNA, and each one of those genes will produce a transcript. And so it's like the expression of that transcript. So it's called gene expression of that gene. So how is this measured? Gene expression is in the lab, and experimentally, it's measured by actually physically counting these transcripts inside a cell. So you would take a cell and you would actually pull out all the RNA transcripts in there, and you would count, okay, I see 5 transcripts of G1, 23 of G3, 20 of Gene 4, and so on. And so essentially you can think of this as like some kind of feature vector for each cell. So each cell has a roughly 20,000 or so protein coding genes, and for each of them you get like a count. And so this vector is usually referred to as the gene expression state of that cell. And so you can get these like very high dimensional vectors for each cell. And so overall you get this nice big matrix that has usually like several thousands of cells and tens of thousands of features. So 2 things to keep in mind about this matrix is about this data is, first of all, the expression of genes in a cell is very important for understanding the identity of that cell, what function it's performing. So it's a really important piece of information and gives you almost all the information you need about the current state of the cell. The other thing to keep in mind is that these genes are not independent of each other. So when you increase the expression of any one of these genes, you also end up usually influencing the expression of many other genes. And so this sort of underlying joint distribution between all of these genes is very interesting for us. And it's also something that we're trying to like approximate using this model. And I'll go into this in the next few slides, but I just wanted to sort of explain some terminology first. So I'll go into the next few slides, but please feel free to interrupt me either through audio or through chat, because I know some of these concepts might be new. So I'm happy to pause and like explain anything. Okay, so what is the actual problem we're trying to solve? So if you find any sort of, if you look at like the key textbook in molecular biology, it actually starts with these few lines where it says that in many respects, we understand the structure of the universe better than the workings of living cells. We still cannot predict how a cell will behave if we mutate a previously unstudied gene. And so this is like very closely related to what we're trying to solve, which is what is the gene expression response of perturbing a gene or a combination of genes that you've not experimentally perturbed. So we want to use machine learning to learn this underlying distribution that relates different genes to each other, so that if we change the expression on any one of those genes, we can predict what happens to the other genes. And this is very powerful, as I'll go on to show you in the next few slides. But that's like the main problem that we're trying to address using graph neural networks. Okay, so let me formally describe this more mathematically so it's clear. So how are we defining a perturbation, and what do we mean by a genetic perturbation? So consider some cells, and so consider a cell which is in an unperturbed state, shown here in green. And so this has some gene expression values, like I showed you before. These have been normalized, so that's why they're floats. And so you can see that it has some expression values for its genes. And so we're applying a perturbation on some subset of these genes. So we're applying it to G2 and G4. And what do we mean by a perturbation? We are like going in and actually using CRISPR and other gene editing technologies. We are increasing the expression of, say, G2, and we're decreasing the expression of G4. So this is what we call a genetic perturbation. And what we want our model to do is to be able to predict the outcome of this perturbation. So we want to predict a simple function. So we want to take as input this unperturbed state X, and we want to take also as input the applied perturbations. So this is, in our model, this is a binary variable that which tells you which genes have been either upregulated or downregulated. And then we want to be able to predict what's going to happen to all the other genes in the cell. So this is the key problem. All right, so why is this relevant? Why do we care? So being able to predict what's going to happen following a perturbation is actually very relevant to a number of biomedical areas. So for instance, consider drug discovery. So if you have knowledge of how a cell will respond if you perturb it, then you could potentially take it from a disease state to a healthy state. So for instance, over here, this is a low dimensional representation of, say, single cell data that I just showed you previously. And so you can see here in blue are what are, like, each dot represents a cell. As you can see, in blue is a trajectory showing healthy cells as they progress through their life. And you can see that in yellow here, we have dots representing cancer, and green, you have some other diseases. And so it would be very useful if we could identify perturbations that would take these cells back from their disease state to a healthy state. And so that's one use for learning about perturbations. Another use could be if we want to understand these underlying sort of interaction effects. So for instance, if we know that knocking out or like turning down the expression of gene A or gene B won't kill, say, a cancer cell, but we want to test if turning them both down will kill the cell, then this kind of interaction is called synthetic lethality, and it's very valuable to be able to predict this. So if our machine learning model can predict how cells will respond to perturbations, maybe it can also predict these interactions, and that would be very valuable. And then finally, if you've heard about recent startups that are trying to think about, like, can we reverse aging, or can we create like artificial T cells that will fight cancer, then those are all doing what is called re-engineering cells. And this is also something that essentially relies on changing the expression of certain genes strategically. So these are like uses for why this would be useful. But then the other question could be, why can't we just do this using experiments? Why do we even need machine learning? The answer to that is that if you think about all possible combinatorial outcomes of perturbations, the space explodes really quickly. So you have like this combinatorial explosion. So even if you just consider all the known protein coding genes, which is roughly 20,000-ish, you have 4 × 10⁸ pairwise combinations of all of those genes. And so that's too expensive and too time consuming for experimentalists to test in the lab. At the moment, most experiments are only looking at, say, a few thousand perturbations at the most. So this is really not feasible. And then if we go even like to higher order combinations, so like if we go to like combinations of 3 genes or more, or 4 genes, then like this goes up by several orders of magnitude. So inevitably, at some point, we'll need to use computation to uncover the space of like combinatorial outcomes of perturbation. And so that's why we think our model would be very useful. Okay, so now let's go a bit more into the method. So this problem is not new, and it's been studied in the past. So in the past, there was a lot of emphasis on trying to understand these relationships between genes in the form of a network. And so this is normally done as shown here. They would try to infer like causal relationships in the form of what they call a gene regulatory network, or you can also just think of it as a directed causal network. But the challenge with learning these networks is that they're very difficult to learn from like large single cell data sets where you have, say, 20,000 variables and hundreds of thousands of observations. And so most existing algorithms don't perform very well. In most cases, they actually fail on such large data sets. And the other issue is that they're not able to capture very well nonlinear relationships. And this might have more to do with existing implementations that don't tend to incorporate nonlinearity. So it's harder for them to capture and a nonlinear effect, which might occur if G1 and G2 are both perturbed in this graph. So some recent approaches have tried to skip this altogether, this network inference step altogether, and they've tried to use, say, like an autoencoder or other latent variable-based methods to go straight from your input to your output. So straight from your applied perturbation and the gene expression and your starting state to what you'd like to be in your final state, without actually inferring this graph between the variables. But then the challenge here is that they don't have an implicit understanding of what these perturbations mean biologically. So if they've not seen a specific perturbation in the past, they don't know how to simulate it. So if they've only seen gene 2 perturbed or gene 4 perturbed, then they won't know what it means if gene 3 is perturbed, because they'll often use like a one-hot encoding. And so the one-hot encoding is entirely dependent on your training data. So they can't sort of expand beyond their training data significantly. And so our approach is to use graph neural networks to sort of combine the best of both of these approaches. So we're still using a deep learning based model. And so this allows us to capture complex relationships between the different genes. But at the same time, we're also able to use our prior knowledge of biology so that if we see a new gene, we can go back to our knowledge graph that is a part of our model and get a sense of like what should happen if that gene is perturbed based on like our knowledge of biology. And so the big advantage of doing this is that we can accurately represent perturbations that were not seen at the time of training. So we can really sort of generalize to new perturbations. All right, so we call this approach graph enhanced gene activation and repression simulator. And for short, we call it GARES. So it's deep learning constrained by prior knowledge of gene-gene relationships. Okay, so in the next 5 slides, I'll walk you through how we sort of built in our biological intuition into our model in the form of inductive biases that helped us to sort of improve the performance of our model. Okay, so these are the 2 inputs that I mentioned previously. And so we have your unperturbed gene expression and you have the applied perturbation. So the first step for us is that we create a multidimensional embedding vector for each gene. And we also create a multidimensional embedding vector for each gene's perturbation. So you have n gene embedding vectors and you have n perturbation embedding vectors. And the reason that we give each gene its own sort of multidimensional embedding as opposed to just a scalar, which is what most existing models do, is because we've seen in the actual perturbation data sets that there's a lot of heterogeneity in how these genes kind of respond, how different genes respond to perturbation. So if you look at the coefficient of variation, it varies a lot in the gene expression response to different perturbations. So we wanted to give the model enough capacity to capture that. The next advantage of using these embedding vectors is that you can kind of simulate the effect of a perturbation within the latent space. So this is akin to, I'm sure most of you have seen the example in NLP literature, where say you have an embedding for king, and you have an embedding for female. And if you add those embeddings together, you can get the word queen. So it's kind of similar to that logic where you have an embedding for each of your genes, and you add to these gene embeddings the embedding for the applied perturbations. So in this case, we're perturbing gene 2, we're perturbing gene 4, and so we add those perturbation embeddings, P2 and P4, to each of the genes. And so this kind of simulates within the latent space the effect of perturbing all the genes with this applied perturbation. The next step for us is to ensure that these embedding vectors are incorporating our prior knowledge. And so we do this using what we call a gene co-expression graph for the gene embeddings. And we use a gene ontology graph for the gene perturbation embeddings. So what do these graphs mean? So in each of these graphs, every node is a gene, and the feature vectors for each of the nodes correspond to these embedding vectors that we had randomly initialized. So here, these embedding vectors are all the gene embedding vectors, and here all the embedding vectors are the perturbation embedding vectors. And what we do is then we use a graph convolutional network. We apply 2 different graph convolutional networks on these 2 networks, and we use those GCNs to enforce that if a specific gene is connected to some neighbors in its graph, then we should enforce that those neighbors are sharing information, and so that their embeddings are somewhat similar within this graph. And so what that allows us to do is that, for instance, if we've never seen G5 before, or if you've never seen a specific perturbation before, then when we predict the effect of that perturbation, then we can sort of rely on the graph to guide us in terms of like figuring out what should be its correct embedding. And then also the biological intuition that we're using here is actually visible also in the actual perturbation data. So if you look at actual single cell perturbation data sets, here you can see different perturbations along the x-axis and the response of different genes along the y-axis. You can see that these patterns are quite visible. So there are many perturbations that tend to produce similar effects, or they refer to these as modules, co-functional modules. And there are also many genes that respond to different perturbations similarly. So you can see like this here, this set of genes responds to perturbations similarly, and these tend to be what are called co-regulated programs. And so we wanted to incorporate this kind of prior knowledge of how these genes are related into our model. And this allows us to like significantly expand our prediction space and also adds additional regularization. All right. So up until this point, we've only been applying perturbation effects at the level of individual genes. So we add in an additional layer, which allows different genes to talk to each other. So we call this the cross-gene MLP layer. And essentially, it's just an encoding or a global encoding of, it takes as input all the different post-perturbation embedding vectors for all the genes, and it creates a summary of that state. And this allows us to account for secondary effects. So for instance, if G9 is affected by the expression of G4, then if P2 has caused P4 to increase in expression, then G9 can use this global state to update its value. So it allows us to account for these secondary effects. And finally, we have the final decoding layer, which is also gene specific, which allows us to capture that heterogeneity I mentioned earlier. And another point to note here is, which is not shown in this graph, is that the embeddings that come out of this step actually are conditioned on this global state. So we don't force all the embeddings to be reconstructed. So this is not like an autoencoder type layer. So every output from every gene has a direct connection to the final decoder, but it's also sort of conditioned on this global state. So this sort of reduces the learning burden on our model. Okay, so this is like the full architecture, and this is trained end to end. These embeddings are randomly initialized, and all the parameters for the graph convolution networks and for all the MLPs here, it's all trained end to end. Now I'll describe to you what the loss is, and also before I describe the loss, the training data used to train this is essentially we have like several thousand cells that have been perturbed in different ways, and we have the actual post-perturbation state for all of those cells. So we can use a simple mean square error to compare the actual post-perturbation state with what we're predicting and update all these parameters accordingly. So let me show you the loss that we use. So this is, if we ignore the autofocus for a second, this is essentially just a mean square error loss. So this is the prediction that we're making for a specific gene, and this is the true expression value for that gene. And so we look at the difference and we square it across all genes, across all cells, and across all perturbations. So one additional factor that we add here is what we call the autofocus loss, which is this gamma term right here. And what this allows us to do is to make sure that the model is not overemphasizing the loss at lower values. So why this is important is because when you perturb a cell, most of the genes are not going to do anything. It's only like maybe a few hundred genes that show most of the effect. And so what we want is that the loss shouldn't be biased by all the zeros. And so we wanted to focus more on like the genes that have been more highly perturbed. So by using this autofocus loss, we can reduce the emphasis that we pay to like these low levels of perturbation and focus more on the high levels of perturbation, which is what is more important to us and to our biologist collaborators. All right, the second thing we add that's maybe a little specific to our use case is a directionality loss. So in the genomics world, it's very important to have an understanding of what is the sign of your gene expression. Like, are you causing the expression to increase or decrease? Sometimes that's more important than the actual magnitude of the change. So for instance, if you have two predictions here, one which is this one here and one which is this over here, and the true value is this blue bar, then both of these predictions would have the same mean square error loss. But for biological purposes, prediction 2 would be more valuable because it's capturing the right direction. And so we incorporate a separate loss term that measures to see whether the predicted sign is different from the true sign of, and by sign we mean the change, the sign of the change in expression over the unperturbed state. And so this is added as an additional term to the loss. Okay, so this is again the final architecture, and you can see here we have again going from the original gene expression and the applied perturbation, we are able to predict what is the effect of that perturbation. All right, so now I'll show you some results. So let's consider here, so these two genes here, P2 and P4, let's consider an example where we're perturbing genes FOSB and CEBPB. So this chart here shows you the effect of this perturbation. So on the y-axis, we see the change in gene expression over control, and control refers to unperturbed cells in their ground state. So after you apply this perturbation, you see that across these many different genes, there is a change in expression taking place. And the change in expression is shown by this black box. So you can see the box represents the post-perturbation state for each of these genes, which are like the most differentially expressed for this particular perturbation class. The green dotted line shows the unperturbed state, and then the red dots here correspond to the prediction that Gears is making. So you can see that Gears is able to quite accurately capture the right trend of post-perturbation gene expression across all of these genes, both in the cases of where we're seeing upregulation of expression, as well as in the cases where we're seeing downregulation of expression. So this is very encouraging. Another point to note here is that this particular gene was not seen experimentally perturbed at the time of training. So in our training set, we had never seen this gene perturbed. So if you were using one of the previous models that used one-hot encodings, they would not have been able to make this prediction. But because we had the prior knowledge incorporated, we were able to make sense of what would happen if CEBPB was perturbed. So we formalized this setting of not seeing a gene perturbed at the time of training more formally. So here you can see 3 different scenarios for what happens when you perturb a combination. So say in your test set, you're perturbing genes X and Y together. Then there are 3 possible scenarios for what might have happened in your training set. You might have seen the gene X perturbed individually and the gene Y perturbed individually. And you're essentially then just predicting what happens when they're both perturbed in the same cell. So this is the simplest generalization case, and we call this 0 unseen of 2. But then there's also the possibility that one of these 2 genes may not have been seen perturbed, so either X or Y, would not have been in your training set. And then you're making a prediction for at least one gene that you've not seen perturbed. So this is like one unseen of two. And then finally, when you may have not seen either X or Y perturbed in your training set, but you're still making the prediction for the combination. So this is the hardest case where you have two unseen of two. So as you go from left to right, you're seeing increasing difficulty of generalization. And so as we see, we looked across the data set that we had, and we found that our model was improving performance across baselines, across all of these three scenarios. We saw an average improvement of about 45% over two baselines. So the first baseline is a linear model, which essentially just uses the co-expression graph and adds some linear weights and then tries to look at the change in expression. And there's also a model called CPA, which is a neural network that it's an autoencoder that does not make use of any prior knowledge. So in compared to both models, you were seeing a significant improvement. The next thing we were interested in seeing is something that I mentioned before, which is the interaction effects. So say you're perturbing two genes, PTPN1, 2, and ZPTB25. It could be this case that the change in gene expression, again, here on the y-axis, is shown here by these two stacked bars, right? So for only perturbing ZBTB25, you might just see this lower stacked bar effect. For this gene, you might see this upper bar effect. And so a naive model may just kind of add these two effects and say, okay, the combination effect is just the sum. But the reality, if you look at the true data, is that it's actually much more than just the sum. There's a synergistic effect. And so what we find is that Gears is able to predict this synergistic effect quite accurately. And so we looked across many other genes in this particular perturbation case where we saw this non-additive effect. And you see that Gears is quite accurately capturing this effect across these different genes. We see this also in the case of like different types of interactions. So here you're seeing that the naive additive model is predicting a higher level than the true data. And Gears is also able to predict this suppressive effect. And you see different kinds of these effects here as well. So this is looking at genetic interaction effects at the level of individual genes, but you can also look at them at the level of the full transcriptome. So for instance, if we consider the effect of a perturbation across all the 5,000 or 10,000 genes to be shown by this vector X, and then that of the perturbation Y be this vector Y, then we can sort of represent the combination as what we call as another vector Z. And so if we were seeing a simple additive effect, then we would just assume that this resultant of X and Y would be the same thing as what we observe actually when we combine these two effects, which is Z. So under a naive additive case, Z would simply be equal to X plus Y. But if you see synergy or suppression or other effects, then Z is not the same as X plus Y. So if you see synergy, then Z is more than X plus Y. So the combination effect is more than the single gene effects. Or in suppression, it's less. And so and so on, you can define these other interaction types. So once we had defined these interaction types, we went back to our data and we tried to predict what are the 10 strongest interactions that we can predict for each of these interaction types. And we compared that to the ground truth to see how well we're doing. So it's the precision at 10 metric. It's like a ranking problem. And we found that our precision at 10 was significantly higher than the existing baselines, with an improvement of like 50% in detecting genetic interactions. So overall, our goal with this model and how we see this practically being applied is that we see many biologists running these experiments for some subset of genes they're interested in and maybe some combinations. And then they can use our model to really fill out the space and make predictions for all the other combinations that are too expensive to run. And so we actually ran this experiment with real data where we trained our model on 236 seen perturbations. And then by using Gears, we were able to make predictions for 5,000 unseen perturbations. And by doing so, we also identified some new phenotypes that had not been seen before. So in summary, I just want to summarize now the talk. We present the first computational approach to reliably predict perturbation outcomes for combinations of genes that have not been seen experimentally perturbed. And Gears is able to uncover substantially larger regions of combinatorial perturbation space, and it can help in the design of new perturbation experiments. So here's a link to the code, and you can also find a link to the preprint in the README for the code. And I want to thank my advisors, Yuri and Steve, and also my collaborator, Kishan, on this project. So thank you for listening, and I'm happy to take any questions.
So for the folks on the call, if you have any questions, feel free to raise it. Okay, going once, twice, 3 times. Awesome. Cool. I think this is a great talk, and I certainly enjoy very much on the talk, and I definitely learned something new. And to be frank, I found the modifying of gene is actually quite interesting, especially in the graph environment. We also are working on this interesting how they're doing the large graphs, especially with the DGL, and also how they internally how AWS is also looking at how they supporting that. So yeah, definitely how they looking forward how they more research how they findings how they from you. Thank you so very much, Yusuf.
Oh, thank you for inviting me. Thank you.
Awesome. Cool. Thank you, folks. If there's no more questions, we're going to end the session short today. And we have actually full pack talk in October. And so unfortunately, today we only have 30 minutes. So thank you so much. And I'm looking forward to see you in our next session. And thank you so much, Yusuf. Thank you all for coming. Bye.